Okay, so now we're going to move into buffers. And a buffer solution is simply a solution that contains an acid-base pair in a relative proportion to resist pH changes. So a buffer solution is a acid-base pair in a solution that can resist pH changes. Oops. And so the pH change can come from an acid or can come from a base. And so buffers are important to keep pH within a narrow and limited pH range, which in our bodies is what's required for all of our enzymes to function properly. So buffers, in essence, are going to resist changes. And keep in mind that carbon dioxide is the largest source of acid in the body. So buffering carbon dioxide is going to be very important. And even though carbon dioxide is a weak acid, it still is the largest amount of acid produced by our cells. Almost every cell in your body will release carbon dioxide when they do cellular respiration, except of course for red blood cells. So a weak acid and weak bases are going to make the best buffers if they're at concentrations that are going to prevent changes in pH at their concentrations. The key is to have a weak acid similar to the pH. So we have our blood pH, and it's approximately 7.4. We have our stomach, and it's approximately 1 to 2 pH. Intestine, pH is approximately 8 to 9. And so changes in 0.2 pH units higher, you're leading into alkalosis, or lower, and you're going more to acidosis. So just some terms: respiratory acidosis leads to a decrease, is caused by a decrease of CO2 exhalation. Metabolic acidosis is going to be a decrease in the bicarb ion. And then respiratory alkalosis is going to lead to an increase, it's caused by an increase of CO2 exhalation. And then metabolic alkalosis is due primarily through excessive vomiting of stomach acid. Okay, so now we have a Henderson-Hasselbalch equation, and the Henderson-Hasselbalch equation is going to help to estimate the pH of a buffer solution. So it's also going to be able to find the equilibrium pH in an acid-base reaction. So it estimates pH of a buffer solution, and it can find the equilibrium pH in an acid-base reaction. Now, the pH, of course, as we know, depends on the amount of hydrogens in the solutions, and we also are aware that strong bases and strong acids dissociate completely, but our weak acids do not. And our pKa is going to measure the ability of weak acids to ionize in a solution. The extent of that impacts the final pH, and so pH and pKa must be related. So again, remember we have our products and reactants in some sort of equilibrium. And then this is given by the equation, where this is an acid, and it's going to be our products over our reactants. And then we're going to take the log of this, and so you get the log Ka is going to equal the log of your hydrogen plus the log of the other two species. So you pull your hydrogen out. And then you can rewrite this as a negative pKa equals a negative pH And then this, of course, you're going to move around negatives, just simple math, and this gives you the Henderson-Hasselbalch equation. So you get your Henderson-Hasselbalch equation. And keep in mind that the pKa is the acid dissociation constant. And pK is basically a quantitative measurement of the strength of an acid in a solution. Okay, so how much does it want to give up? So, and remember, the strong acid has the lower pKa. And it's opposite for Ka, obviously. A stronger acid has a higher Ka. Okay, so the Henderson-Hasselbalch equation can be turned into a graph, and it can be used to determine the amount of acid and conjugate base needed to make a buffer solution at a certain pH. So here we have our pKa, where we're in equilibrium at a certain pH. And then as you add more base, so as you add more base, you're going to exhaust the conjugate acid. And as you add more acid, you're going to exhaust the conjugate base. And so basically, a buffer can really only protect with a +1 and a -1 pH range. So if you have a buffer that works best as a pH of 7, it can buffer between 8 and 6. So it'll keep that pH around 7 for a period of time. But once you get beyond those +1 and -1s, it is no longer able to provide any buffering capabilities, and the pH changes dependent on the addition of the acid, or in the case of it rising, the addition of the base. Keep in mind a few things. So it's rare that pH equals pKa. And if pH equals pKa, that does not mean a neutral pH, because pH is always simply a function of how much hydrogen is actually in there. So the pKa is the pH value at which a chemical species will accept or donate a hydrogen. Let me write this here. Oops. Okay. The lower the pKa, we know, is the stronger the acid with the greater ability to donate a proton. Okay, so now let's talk a little bit about our buffers. So our buffers are going to be solutions of weak acid and their conjugate bases, and these are going to resist a pH change ±1 of their pKa, basically. Keep in mind that there are some rules. So the greater the concentration of the conjugate acid and the conjugate base, the greater the buffering capability. But it's not limitless. So for example, if a solution of your conjugate acid and conjugate base is equal to 1, that will have a greater buffering capability than the same conjugate acid and conjugate base equal to 0.1. You just have more of the species present to perform buffering. So in the case if you had the conjugate acid equal 1 and then you had the conjugate base equal 0.1, in this particular buffer you will have a higher capacity for addition of a base since you have more acid present. Okay, so you just simply have more of your conjugate acid present to buffer the addition of a base, rather than addition of an acid will overwhelm the conjugate base. So here we have acetic acid, which is a weak acid, and it's going to become acetate. And then if you're going to add hydrogen chloride, the species will generator will react with the acetate ions. And then the buffering capabilities have to be flexible, and the flexibility is that the conjugate acid and base should be as close to equilibrium with each other as possible. And this provides flexibility in buffering. Now, of course, again, buffers cannot buffer indefinitely. They can only protect with the pH within 1 pH unit of pKa. So again, that's going to be, if you're a good buffer at a pH of 9, you can buffer between 8 and 10. If you're a good buffer at a pH of 2, you can buffer between 1 and 3. Now, what's the importance of buffers? Well, it has to do, especially in our bodies, has to do with our proteins. So this is a protein. All of our enzymes are protein. And so in order for our body to function, we have to be at a physiologic pH. And this makes the active site available to perform some function. If you lower the pH, you're going to denature the protein, which means it falls apart. And in essence, you have no activity because you lose that active site. When you go back to a normal pH, you can renature and refold that protein, and so now you gain that active site back. Okay, so let's talk a little bit about metabolic acids and buffers. So again, we have some pHs that are important for the body. The blood pH is about 7.4. Intracellular pH is between 6.9 and 7.4. The maximum pH range inside of your body is 6.8 to 7.8. Acid is always going to be excreted. So we have CO2, which is going to be exhaled. You will have urine, which is going to be excreted. And then if you do have acids in your body that cannot be excreted, they have to be buffered. And so we have some buffering systems. We have a bicarbonate buffering system, and this is the most important. Extracellular. And then we have the hemoglobin buffering system in red blood cells, and this is going to be able to pick up hydrogens before they change the pH of the blood. We have the phosphate buffering system in all cells, and this is the most important. Intracellular. And we have a protein buffering system of both cells and plasma. So this is both intra plus extracellular. And I'll put here important to buffer blood pH. Okay, so let's talk about the bicarb system pretty quickly. So again, CO2 is a major source of acid in the body. CO2 is going to dissolve. It's going to produce carbonic acid. Carbonic acid is going to be be generated from carbonic anhydrase. Carbonic anhydrase is in abundance in your red blood cells, so it does this pretty quickly. Carbonic acid and bicarbonate are going to be that conjugate acid-base pair. And so one of the things to keep in mind is carbonic acid has a pKa of 3.8. And because of this, our blood pH is at a 7.4, so carbonic acid is going to be completely dissociated. And so you're going to get bicarbonate. Now, CO2 is the source of acid in the body, but carbonic acid is the most abundant acid in the body because it's what is generated from CO2 via carbonic anhydrase. Now, one of the things to keep in mind is that our respiratory from our cells and releasing CO2 is always going to be at equilibrium with the exhalation of CO2 in the lungs. And of course, breathing can increase or decrease CO2 levels. So if you decrease breathing, you're going to increase CO2 in the blood, which makes it more acidic. If you increase breathing, you're going to decrease CO2 in the blood and make it more alkaline. All right, so our bicarb buffering system is basically the main extracellular buffering system to eliminate metabolic CO2. And so this really is three reactions. We have, of course, our CO2 released by our cells, and we have our aqueous environment, which is in our blood. And then this is going to become our carbonic acid by that carbonic anhydrase. And then this, because of the pH of our blood, is going to quickly dissociate into bicarbonate. And then this is going to be our acid-base pair. Now, when bicarbonate is going to be released into our blood, there's hydrogen in the blood that'll quickly convert it back to carbonic acid. And carbonic acid is going to be favored in an aqueous environment 20-1. Now, CO2 is also, as I said before, going to be expressed as a fraction of partial pressure CO2 in arterial blood, pressure arterial CO2. And the buffering system is going to be coupled to the removal of CO2 from the blood as well as exhalation from the lungs. So the blood buffering system is coupled to the removal. So in essence, CO2 in the blood is going to be at equilibrium with CO2 in the lungs for exhalation. All right, so the pKa of our bicarbonate breaking down is going to be 9.8. So this hydrogen bicarbonate and CO3 minus 2 acid-base pair is not physiological favored in our bodies. So there's not going to be much of that. Instead, it's going to be this reaction up here that's going to be more favored in our body. So this is the bicarbonate buffering system. You have reaction 1, in which you have carbonic acid, and bicarbonate is the proton acceptor. Reaction 2 is where you have our carbonic acid being formed from dissolved CO2 and water, and then in reaction 3, you're going to have your carbon dioxide in aqueous solution be in equilibrium with the gas phase in the actual lungs. So let's just talk a little bit about buffering in the red blood cell. So again, bicarbonate, carbonic acid are going to be the major buffers for the plasma as well as interstitial fluids. You first have your carbon dioxide, which is a waste product. And it's a weak acid. Okay, and so you're going to release carbon dioxide. Carbon dioxide is going to be picked up by your red blood cells. Red blood cells have a large amount of an enzyme called carbonic anhydrase. This is going to form carbonic acid. This will dissociate into bicarbonate and a hydrogen ion. The hydrogen ions are going to be buffered in the red blood cell by hemoglobin, or by our phosphates. Phosphates are another important buffering system. And then your bicarb is going to be exchanged 1-for-1 with chloride in your red blood cell. It'll be released into the blood where it will bind to a hydrogen, and it will be transmitted through the blood as carbonic acid. Now, just as an overview of this system, you have, for example, your hepatocytes, and they are going to have mitochondria. It's not nucleus, this is mitochondria. And they are going to do oxidative phosphorylation, and they're going to release CO2. And this is going to be constant, of course. And then in your red blood cell, so very large red blood cell, not to scale. Hepatocytes are bigger. Okay. The red blood cell is going to pick up all of these CO2s. And with the carbon anhydrase, going to make your carbonic acid inside of the red blood cell. Let's see. And this will quickly dissociate into a hydrogen and your bicarb. And then there is a receptor, an exchange receptor on the surface of your red blood cell, where every time you have a bicarb made, it will leave. And it is in exchange for one chloride ion coming in. So it's a one-to-one exchange. And then your hydrogen is going to be buffered by your hemoglobin. Or by a phosphate. Okay. Now, this is happening in the tissues. So over here is in our tissues. And then everything enters the blood. And eventually it makes its way to the lungs. And in the lungs, you have your alveoli. Just make them like little. Oops. And so what happens here is your red blood cell comes over. So here's your red blood cell again, not to scale. And your bicarb becomes carbonic acid. And then in the lungs, this is going to again exchange one-to-one with chloride. So again, this is a one-to-one exchange. And now you're going to have your carbonic acid once again inside of the cell, which is not going to be favored. And so this is going to be broken down by that same enzyme, that anhydrase. And you're going to get CO2 plus water. And then the CO2 is going to be exhaled and exchanged for oxygen. And then oxygen is going to come in. And we had our hemoglobin, which released its hydrogen. And it became hemoglobin, so now it can pick up the oxygen in the lungs. And so that is how you're going to exchange carbon dioxide made in the tissues for oxygen in the lungs. Now, the bicarb buffering system is also very important for the kidneys. So the kidneys have a role in all of this too. So again, in the lungs, we had over here our H2O plus CO2. CO2 is obviously an acid. We're going to exchange that for oxygen. And then we're going to have it at equilibrium with our carbonic acid in the kidneys. So what's happening in the carbonic acid in the kidneys is this, again, is going to be at equilibrium with our bicarbonate, which is our base. And then this is going to have, of course, our hydrogen, which is an acid. And this acid gets excreted in the kidneys. And our bicarbonate, gets released into the blood, plus a hydrogen. And then you go back to our carbonic acid, and this can go to the lungs or I can go back to the kidney. So the kidney is going to help to remove excess hydrogens, excess acid through excretion. So our bicarbonate levels are going to denote a metabolic kidney effect. So elevated bicarbonate raises pH, and then low bicarbonate is a sign of metabolic acidosis. And so metabolic acidosis can be caused by many things. So its causes are cancer, carbon monoxide poisoning. You can have alcoholism. You can have liver failure, hypoglycemia. And some medications. Okay, so even though our carbonic acid is our most abundant acid, there are other metabolic acids that are important. We have our ketone bodies that can become acidic, especially in type 1 diabetics. So our two ketone bodies that we can use are acetoacetic acid, as well as beta hydroxybutyrate, and acetone, of course, gets exhaled. You can have lactic acid, which was going to be caused by glycolysis in muscle cells, red blood cells. Some things to note that if cells become too acidic, hydrogen will exchange for sodium. And remember, hydrogen can be brought to the kidneys and it can be excreted. And so that's a way to get rid of excess hydrogen. If cells become too basic, your bicarb will exchange for chloride. So remember, sodium chloride is very important, extracellular. And so having those two ions available, it can help to balance acid bases inside of the body. Urinary acid is a non-volatile acid. And it's going to be excreted by the kidneys. Acid that's going to also be secreted come from different sources: uric acid, citric acid from TCA cycles, sulfuric acid, etc. pH of your urinary pH is 5.5 to 7.0, and the minimum is 5.0, pH of 5.0. Now, ammonium is going to be important as a buffer of the urine. So our ammonia is toxic. It has a pKa of 9.2, so the ammonium ion is favored at a pH of 7. So ammonium is going to buffer urine pH. It's going to be released in amino acid catabolism when you break down your proteins and amino acids. The ammonium is also going to be toxic to neural tissue. Your kidneys are going to excrete the ammonium ion in proportion to the acidity of the blood, how much hydrogen is being delivered via the carbonic acid to the kidneys. And the renal tubules are what are going to transport that excess hydrogen that was brought by carbonic acid to the urine, and it will return those bicarbonate anions to the blood to pick up more hydrogen. All right, so overview of buffering systems. So buffers are going to maintain body pH. There are three major. We have the bicarb, carbonic acid system. We have our phosphate system, and we have a protein. And proteins are going to be hemoglobin in RBC, and any amino acid with a histidine. Yes, amino acid with are going to be excellent buffers. So in general, we have our intracellular fluid, which is going to be buffered mostly by that phosphate system. And then by proteins, we have our extracellular fluid, critical for carbonic acid. And again, the carbonic acid, remember, is going to be bicarbonate picking up hydrogens and then taking them to the kidney, or the bicarbonate picking up hydrogens and taking carbon dioxide to the lungs. Hemoglobin buffering system is important for red blood cells. And again, it's because red blood cells have that carbonic anhydrase, so they're going to convert CO2 and water to carbonic acid, and carbonic acid inside of the red blood cell will dissociate and release hydrogen ions, which need to be buffered, and so they get buffered by the hemoglobin system or the phosphate system. And proteins are going to be both intracellular and extracellular, so they are going to be able to buffer both. Okay, so hydrochloric acid, gastric acid, and keep in mind stomach pH is about 1 to 2. It's going to be secreted by our parietal cells. It is important for denaturing proteins. And keep in mind the small intestine has a pH of 8 to 9. And so when gastric acid enters the small intestine, you have your pancreas epithelial cells, and they will secrete bicarbonate, and this will neutralize your gastric acid as it enters the intestine. Now, just really quickly, some diseases associated with pH balance or imbalance, type 1 diabetes and ketoacidosis. So type 1 diabetes, you have no beta cells in your pancreas. It's an autoimmune disease. And you have no insulin. And no insulin means that, for example, your liver is in constant fasting stage. And so regardless of your food intake, your liver thinks that it's fasting, so your liver is constantly making its own glucose through gluconeogenesis. It's constantly making ketone bodies. Even if you have an increase of insulin, your muscle cells have no insulin to tell them, so they are not using that glucose that you just ate. And so, again, you're in an untreated type 1 diabetic, your body believes that it's constantly fasting, regardless of your intake of food. Now, when you have an aspirin overdose, this can cause hypothermia, because aspirin is what is referred to as an uncoupler of oxidative phosphorylation. Basically, the electron transport chains, and all your cells get shut down. And so this is not hypo. Um, uncoupling of your inner mitochondrial membrane is going to lead to the production of heat, and so you have hyperthermia. Also, if by uncoupling oxidative phosphorylation, you're going to push for anaerobic glycolysis. So you have an increase of lactic acid. You also are going to increase ketone bodies. So you have an increase of ketone. And so you become more acidic. And then in response, your patient will increase respiration. And so this is going to lead to respiratory alkalosis. And so you have these two things trying to counter each other. You have the increase of acidity, which is caused by the decoupling of the oxidative phosphorylation and pushing everything towards anaerobic. And that is going to try to be countered by your respiratory rate, which becomes now more alkaline. So you have an acidic blood and alkaline respiratory situation. And so you're going to have a major changes in your acid-base management within the patient. And of course, hyperventilation is going to lead to alkalosis. And again, it has a similar acid-base management impact.